My dear students, welcome to my today's online lecture class, continuation of topics of recombinant DNA technology. We have learned so far overall view of what happens in recombinant DNA technology experiments. Then you have learnt about molecular tools, that is restriction endonucleases and DNA ligases and alkaline phosphatase. Knowing about enzymes is one requirement to conduct recombinant DNA experiments, but apart from enzymes, you have to know so many other factors which are needed for the successful completion of an RDT experiment. So, I am Dr. MEERA B K, Associate Professor and Head, Department of Zoology, Maharani Science College for Women, Palace Road, Bangalore 1. You can see my cell phone number and email ID on the slide. After the talk, if you have any queries, any questions, do contact me either over my cell phone or to my email ID. Now, let us know about vectors, another important tool that is needed to conduct recombinant DNA technology after learning ourselves about the various types of enzymes that are needed. Now, what is a vector? Vector is basically a Latin word. The etymology goes like this: vihir. Vihir means to carry. So, vector means a carrier. You must have studied as zoology students vectors in parasitology, but there vectors is altogether a different aspect. In recombinant DNA technology, though they are used in the same sense, that is as carriers, their role to be played is totally different. What is the definition then? Here in RDT, a vector is a DNA molecule that has the ability to replicate in an appropriate host cell and into which the DNA fragment to be cloned is integrated for cloning. Here you should note two things. One is a vector should have the ability to replicate in an appropriate host, whether it is a bacterial cell or a plant cell or an animal cell. And one more thing is it should be able to successfully integrate the DNA fragment of our choice in which we are interested to bring about gene cloning. So, a vector is a DNA molecule that has the ability to replicate in an appropriate host cell and into which the DNA fragment to be cloned, what we call DNA insert, is integrated for cloning. Now, please note, vectors as such do not exist in nature. We have to design our own suitable vector for a particular experiment in the laboratory, picking up so many components from different sources. Okay? So, please note, none of the vectors exist naturally. Okay? They are all designed so as to suit to our purpose of experiment. Now, what are the characters of an ideal vector? If I want to have a vector, what are all the characters I expect to be present in it? One is they should be capable of autonomous replication. They should be able to replicate on themselves. When it is possible, only when they have their own ori of replication, origin of replication. So, this topic needs a little bit background of molecular biology Hope you have studied in your previous semesters. So first thing, it should be capable of autonomous replication. That means a vector should have its own ORI of replication. Then it should be easy to isolate and purify. It should be easy to isolate and purify. This is one more character what I expect in vectors. Then a good vector should enable easy transformation. A good vector should enable easy transformation. What do I mean by transformation here? Transformation is a process of gene transfer by which some bacteria take up foreign genetic material. I repeat, it is not there on the slide. Please note down. Transformation is a process of gene transfer by which some bacteria take up foreign genetic material. Okay. So a good vector should enable easy transformation. It should get transformed easily. Then should have suitable marker genes. Should have suitable marker genes so that screening and selection of the transformed host cells will be easy. See, screening and selection of the recombinant host cells is altogether a big topic in RDT, altogether a different chapter where there are different methods, and unfortunately it is not covered in the syllabus. But right now you just know they should have a suitable marker genes so that selection and screening is easy. What do I exactly mean by a suitable marker gene? A suitable selectable marker is a gene introduced into a cell, especially a bacterium or the cells in culture, that confers a trait suitable for artificial selection. That confers a trait for artificial selection. There are different methods like insertional inactivation, alpha complementation, immunoprecipitation, immunological methods, etc. But you are not studying that aspect in detail in this paper. Okay? Now, should contain unique target sites for as many as restriction enzymes as possible. Should contain unique target sites for as many as REs as possible. So here, restriction enzymes I have taught you already, and by now you must be knowing what are target sites. So these are all the properties, good properties that I expect to be present in an ideal vector. Now, after knowing the properties of a good vector, you should know the types of vectors. Normally, we study two types of vectors in RDT. One is cloning vector, another one is expression vector. What are they? Let me tell you. One is cloning vector in the sense, these are small DNA vehicles that carry a foreign DNA fragment. Foreign DNA fragment. Okay. And here the purpose of designing such a vector is just to propagate the DNA of your choice. Like you want to have multiple copies of the DNA insert. Okay. In such cases you will use cloning vectors. So the main purpose is propagation. Once you get multiple copies, you may use them for different purposes like research, for further studies and other things, right? Here expression is not the motto. The retrieval of proteins is not the motto. So it is a small DNA vehicle that carries a foreign DNA fragment. Best example for this are plasmids as vectors and lambda phages. That is a type of bacteriophage, right? Now, we are studying mainly cloning vectors as per your syllabus. Then the second type is expression vectors. It is a relatively small DNA molecule used to carry a specific gene into a target cell. Used to carry a specific gene into a target cell. Inside the host cell, the protein that is coded for by the gene is produced. So here the motto is to produce the proteins that is coded by the DNA of your choice. Okay, by regular transcription and translation processes of the host cell. So in cloning, getting multiple copies is the motto. In expression vectors, retrieving the protein, required protein is the motto. So there are two types of vectors which are normally used in RDT. Okay, but we are studying only about cloning vectors. All right. Hope it's clear. Then, depending upon the host, vectors are cloned into which the vectors are cloned. Okay. Expression vectors are classified into two types. Say for example, if you put a expression vector into a prokaryotic cell host, we call them as prokaryotic vectors. On the other hand, if you put them in an eukaryotic cell, host cell, we call them as eukaryotic vectors. Okay. So after knowing about the definition, properties of a good vector and the types of vectors that which we normally use in RDT, now we should study different types of vectors wherein according to your syllabus you are studying only three. One is plasmid vectors, another one is bacteriophage vectors, another one is Classmates. Okay. Now in this class we will study about plasmid vectors. Rest of the two we will be studying in our next class. Now coming to the plasmid vectors. What are they? Before going into the vectors as such, you should know what are plasmids. Plasmids are extrachromosomal DNA elements capable of independent replication and propagation. The whole genetic unit containing the ore of replication and its associated control elements is called a replicon. So another teacher will be teaching you about recon, muton, etc. in another genetics class, but right now we just know what is a replicon. So plasmids are extrachromosomal DNA elements capable of independent replication and propagation, and that entire unit containing the ore of replication and relevant associated control elements is called a replicon. Now, after knowing the properties of an ideal vector in general, now you should know the properties of a good plasmid vector. Number one, smallest possible so that it is easy for isolation and handling. That's the only purpose why you prefer smaller ones. Then they should be capable of autonomous replication. Okay. Then they should contain single restriction site for as many as restriction enzymes possible. Okay. Then they should have clear selection markers. Just now I have taught you this to confer a selective phenotype on transformed cells. I will tell you a little later what exactly this is. Then restriction site should be available at least in one of the marker genes. When I take up the example of PBR322, I will explain what exactly I meant. Then for biosafety purposes, please note they should not be conjugative. They should not be conjugative because sometimes, if they are conjugate and by chance, if they are released into the environment, it becomes very hostile. So for biosafety purposes, they should not be conjugative. Now, one best example for plasmid vector is PBR322, with which all of you are familiar. Now, PBR322, what it stands for? P, it stands for plasmid. It tells you that it's a plasmid vector. BR stands for the people who developed it, Bolivar and Rodriguez, who developed the plasmid, and 322 stands for plasmid serial number. Okay, then yeah, this is the picture of the PBR 322, okay, which is an Escherichia coli cloning vector. That means you can clone into this E. coli cell. Host cell is the Escherichia coli. Now let us see the properties of this PBR 322. It is 4.3 kb Daltons. It contains 4.3 kb base pairs. Okay. Then 4.3 kb base pairs. Then it contains ORI, origin of replication, which is derived from PMB1, So the ORI is derived from PMB1. Then second one, it has tetracycline resistance gene. You can make out here in the diagram tetracycline resistant gene, the almost pink colored structure, tetracycline resistance gene. This is derived from PSC 101. Students, I have already told you none of the vectors exist naturally. You have to pick up different elements from different sources and design the vector of your choice. So ORI is derived from PMB1. Then tetracycline resistance is derived from PSC 101. Then ampicillin resistance gene, which is there right opposite to tetracycline resistant gene, that is derived from TN3. TN3 is a part of R1DRD90. Okay, then it is one of the standard cloning vectors and is the grandparent of many other vectors. So many other plasmid vectors have been derived from this PBR 322. So you can see it has molecular weight of 2.6 Daltons and it has 4.3 kbp, and then you can make out in the tetracycline resistance gene region there is a site for BAMHI, SPH1, SAL1. On the other hand, in ampicillin resistance gene there is a site for SCA1, PST1, and then next to that you have SSP1 region, HIND3 region, in between you have EcoR1 target site region, and then below tetracycline resistance gene you have PUV site, and then adjacent to that you have ORI of replication. So this is how PBR 322 looks like. Then the advantage of this vector is there are over 40 enzymes. There are over 40 enzymes with unique cleavage sites. 40 enzymes with unique cleavage sites, and out of 40, 11 of them are in tetracycline resistance gene and 6 of them are in ampicillin resistant gene. Okay? Hence, cloning, when if you want to clone the DNA of your choice, it can be done using these 17 enzymes as they result in insertional inactivation of one of the marker genes. Okay? Either the ampicillin resistance gene or the tetracycline resistance gene. Okay? This can be used to select the transformed cells. Now, I have used the term insertional inactivation. It is one of the methods normally used for screening and selection of the recombinant colonies. What exactly I mean? Note down, it is not there in the slide. Insertional inactivation is a technique used in RDT. The bacteria carrying recombinant plasmids or a fragment of foreign DNA is made to insert into a restriction site inside a gene to resist antibiotics, thus causing the gene to turn non-functional or to turn into an inactivated state. Say for example, see here you can see the tetracycline resistant gene, right? If you have a DNA of your choice, and if you cut here by making use of BAM-HI and insert the DNA of your choice, the DNA insert will sit in between the tetracycline resistant gene. When it happens, this tetracycline resistant gene becomes inactive. It becomes inactive. By inserting the DNA of your choice, you are making tetracycline resistant gene inactive so that it will be susceptible to that particular antibiotic. So that screening and selection will be very easy, right? So selection of the transformants is by insertional inactivation, and that is followed by replica plating method. Okay. So children, so far I have taught you about vectors, properties of a vector, types of vectors, and one particular type of vector, that is plasmid vector, with one example. Thank you for your patient hearing. Stay blessed. Thank you. Hello, my dear students. Welcome to my today's online class of recombinant DNA technology. Continuation of vectors. Vectors part 2. I am Dr. MEERA B K, Associate Professor and Head, Department of Zoology, Maharani Science College for Women, Palace Road, Bengaluru 1. You can see my cell phone number and so also my mail ID on the slide. After the class, if any queries, do contact me through these venues. Now let us start our class on vectors. In my previous class, I had taught you about vectors, types of vectors, and I had discussed in detail about plasmid vectors. Now, in this class, we are going to learn two more types of vectors. One bacteriophage vectors and the other one Classmates. In bacteriophage vectors, we are going to learn about lambda phages as vectors. Okay? By now, you must be knowing that bacteriophage is a virus whose host cell is bacteria, right? So, we are going to learn about phages which have been exploited as vectors. One such thing is lambda phage vectors. Okay? Now, what are phages? Phages are the viruses that replicate within the bacteria. The advantage of these phage vectors compared to the plasmid vectors is that they can take up larger DNA fragments. They can take up larger DNA fragments when compared to plasmids. So that if the size of your DNA insert is bigger, then you can always go for phage vectors than plasmid vectors. Okay? Now, you have to do some background exercise for this. Open your microbiology textbooks. Make a note on biology of lambda phage. Here I am just going to brief you. Lambda phage is a virus of E. coli, that is Escherichia coli, and it has been most thoroughly studied. Okay? Since we know the biology very well, people have attempted to develop that into a vector. Its biology goes like this. It has a molecular weight of 31 into 10 to the power of 6 Daltons, and the genome has 4805 base pairs. So the length goes as 4805 base pairs. Now, this lambda phage DNA molecule size can be increased only by 5% of the new DNA. Like if you want to retain the entire genome of lambda phage as such, plus you want to add DNA of your insert, it should not exceed by 5%. That means it comes to around 3 kb, because if the total size is more than 52 kb, as such now the size is 48 kb, if the total size becomes more than 52 kb, the packaging of the viral particle into the head becomes a problem. Now you have to learn about biology of lambda phage vector as homework. Okay. So 48 plus 3, 51 or 52 kb. Conveniently it can be packaged into lambda head structure. So if you are not removing any genomic part of lambda phage, there is scope to add only 3%. 5% of extra DNA, that comes to 3 kb of new DNA. Okay. Now, the idea of developing beta vectors with lambda phage came in the way with the discovery that lambda segments could be deleted, lambda DNA segments could be deleted without impairing viability. What I meant is, out of that 4800 and odd kb, if you remove part of it, then also lambda phage will be normal only. Okay. So people came up with the idea that by deleting non-essential regions of the genome, you will have more scope to add the bigger DNA insert. This made people to work on how to develop lambda phages into vectors. Okay. Now, removal of all or part of this non-essential region. You know, the non-essential region lies between the positions 20 and 35 of the genetic map. Okay. So, if you remove part or total of this region of the genome between 20 and 35, this decreases the size of the molecule by 15 kb. So, here you will get around 15 kb, and as such you can add 3 kb. That means as much as 18 kb of new DNA can be added, okay, without hampering the package process of lambda phage heads. Okay. And the lambda genome is so large that it has more than one recognition sequence for almost every restriction endonuclease. Okay. So this made people to exploit lambda phages as vectors. Okay. Now, they have designed so far two types of lambda phage vectors. One is insertion vector and the other one is replacement vector. We shall study both. Now coming to insertion vectors. Here vectors have a single target site for the insertion of foreign DNA. Okay, for the insertion of foreign DNA, and wherever there is a single target site, just a cut is made and in that you will insert the DNA. Such vectors are known as insertion vectors. What you do is, a large segment of this non-essential region is deleted and the two arms are ligated. When you do so, the genome size decreases by 20%. Okay. So such phages will have 20% less than the wild phage DNA. And they possess at least one unique restriction site into which you can insert DNA of your choice. The size of the inserted DNA fragment that an individual vector carry depends upon the extension to which non-essential region has been deleted. Okay, non-essential region has been deleted. Now you can see in the diagram, the first one is the insertion vector. You can see the linear molecule of lambda phage. On either side, it is flanked by cososites or cohesive termini, what we call. And in insertion vectors diagram, there is one place for eco R1, that is the place where there is target sequence for eco R1. What is done is, this linear DNA molecule is given to eco R1, a cut is made, and the DNA of your insert, DNA of your choice is inserted in this region. Okay. How exactly it is done, the diagrammatic representation I have given in the notes, you can just refer. Okay. Now, best example for this type of insertion vector is lambda GT10. I'll come to this slide later again. Best example for insertion vector is lambda GT10. This can carry up to 8 kb of new DNA, which is inserted into unique eco R1 site, and this site is located in C1G at the position 857. So insertional inactivation of this gene means recombinant molecules can be distinguished by clear plaques. So what is done in lambda GT? You will take the linear DNA molecule, you will identify the non-essential region. In that C1 region at the position 857, which contains the target site for eco R1, there a cut is made, digestion is brought about with the restriction enzyme eco R1, and then in that place you will insert the DNA of your choice. Up to 8 kb of new DNA insert can be put in insertion vectors. This is what I wanted to tell you about insertion vectors. Coming back to the slide, this is the place where the place of eco R1 where the cut can be made and your DNA can be inserted. Then people have designed another type of vectors making use of lambda phage only. They are called replacement vectors. You can make out in the diagram, the replacement vectors have two recognition sites. They will have two recognition sites instead of one as you see in insertion vectors. So you will have two sites for cloning. You can see here the linear DNA molecule with cohesive termini at both the ends. You have two positions of eco R1 where you find the target site for eco R1. In between, there is a DNA segment measuring up to 20 kb. Now, what exactly is done is, this molecule is given with eco R1 and given for digestion. So, there are two cuts being produced, and that 20 kb of non-essential region of the genome of phage DNA is removed. In that place, you can add the DNA insert of your choice. So it goes without telling that you can insert DNA inserts of bigger size in replacement vectors when compared to insertion vectors. Okay. So up to 24 kb you can insert. Best example for replacement vector is lambda EMBL4. Lambda EMBL4, the bottom one. I have given the diagrammatic representation of how exactly insertion is done in my notes. Please refer to my notes. So EMBL4 can carry up to 20 kb insert DNA by replacing a segment flanked by pairs of eco R1. Sometimes BAMH1 or SAL1, any one of these restriction enzymes can be used. Okay. Here in the diagram I have shown the eco R1 sites. And selection and screening is not a problem at all. Recombinant selection with lambda EMBL4 can be done on the basis of size, or you can utilize the SPI phenotype. You can utilize the concept of SPI phenotype, SPI. I will tell you what it is. Now, so same thing, I have put it here, the details. Now, selection by using SPI. What exactly it is? See, phages cannot infect Escherichia coli cells which are already infected or which already possess an integrated form of a related phage called P2. Say, if an Escherichia coli is infected by P2 phage, such E. coli cannot be infected by the lambda phage DNA, and therefore they are said to be SPI. SPI means sensitive to P2 inhibition. SPI means sensitive to P2 inhibition. But some lambda cloning vectors are designed so that insertion of new DNA causes a change of the E. coli from SPI plus to SPI minus. SPI plus to SPI minus. See, to begin with, if the E. coli cell is SPI plus, when you insert this vector with the DNA insert, the E. coli will become SPI plus to SPI minus. It will not be sensitive to P2 inhibition, and therefore in such cases selection of recombinant cells is very easy, because recombinant cells to infect that carry P2 prophages. Okay, so such change from SPI plus to SPI minus will enable recombinant cells to infect cells that carry P2 prophage. Okay, so such cells are used as host cells for cloning experiments with these vectors. Only recombinant cells are SPI minus, whereas the others, all non-recombinant cells, will become SPI plus, so that only recombinants form the plaques. So selection is by plaque formation. Okay. So this is about the replacement vectors, and best example is lambda EMBL4. What are the advantages of phage vectors over plasmid vectors? These are more efficient than plasmid vectors for cloning of DNA fragments. The largest insert size can be 24 kb in lambda phage vectors, while that for plasmid vectors it is less than 15 kb. This is one advantage. So insertion of large DNA inserts possible. Then it is easier to screen a large number of phage plaques. So in a culture plate than bacterial colonies for the identification of recombinant vectors. So two advantages are insertion of DNA of larger segment up to 24 kb instead of 15 kb. Then easy selection method. Okay. These are the two advantages of phage vectors when compared to plasmid vectors. Okay. Now, this is about phage vectors. Here I have given a table which tells you the differences or comparisons between insertion vectors and replacement vectors. Two key differences are: insertion vector is a type of phage vector in which a unique restriction site is introduced within the phage genome at the site of optional DNA. Okay. Whereas in replacement vector, here vector is developed from the removal of a middle filler pigment region of the phage DNA. One difference. Second difference, the replacement vectors accommodate higher lengths of foreign DNA inserts when compared to insertion vectors. Okay, these are the two differences. Then, as per your syllabus, you have to learn one more type of vectors, that is Classmates. Classmates, these are the hybrids between a phage DNA molecule and a bacterial plasmid. Cos is from phage part, MID is from plasmid. So, it is a hybrid of plasmid and a phage vector. Okay. So, hence the name Classmates. The design centers on the fact that, packaging of lambda DNA molecule into the phage protein coat need only the cause sites. Cause sites means cohesive termini in order to function. So, what all is needed is just the cause sites from lambda phage. Okay. So, that means the in vitro packaging reaction works like this. Not only for the phage genomes, but with any molecule that carries cause sites on either sides separated by 37 to 52 kb of DNA. So, what is needed from phage DNA is just the cause sites. Okay, what exactly it is, I will tell you a little later. Now, so you retain the cause sites and few other segments from lambda phage DNA, and in between you can add the DNA of your choice, and the formation of head or packaging takes place as usual. It is not hampered. It doesn't require only the genome. Instead of genome, any molecule that carries cause sites separated by 37 to 52 kb. The insert should not be more than this much. Okay. As usual, it is packed. Right. So, such a vector is known as Classmate. Now, what all the Classmates contain? Classmates essentially contain 250 base pairs of lambda DNA. 250 base pairs of lambda DNA. What all it includes? One, I have been telling you the cos site, COS site, cos site or cohesive termini. What exactly it is? Okay, cohesive termini from 5' end to 3' end. It runs like this: G, 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 C, G, G, C, G, A, C, C, T. This on either side single-stranded molecule. Okay, single-stranded molecule. So, these cohesive termini, also referred to as cause sites, allow the DNA to be circularized after infection of the host cell, and cause site helps in circularization of DNA and plays an important role in packaging. So, this part of lambda phage DNA is retained. Okay. Then, second thing, sequences needed for binding and cleavage by terminase. Sequences needed for binding and cleavage by terminase, so that under appropriate conditions they are packed in vitro into empty phage particles. Alright. So, these are the two things that are needed from lambda phage DNA. Then rest you can take from plasmids. Okay. Now, a typical Classmate should have one ori of replication because autonomous replication is needed. Then it should have unique restriction sites. Then it should have selectable markers from plasmid. Selectable markers from plasmid and the cause site. Alright. So, these are the 4 requirements that a typical Classmate vector should have. Okay. Then, here I have shown you the structure of a Classmate. Classmate. Structure of a Classmate. Right. So, this Classmate vector is known as PJB8. It has 5.4 kb length, and you can see here one ampicillin resistance gene, and most important is Cas site, right? Cas site, and then there is one region for Hint3 and Sol1, and whatever is marked here with yellow, only that region has been taken from lambda phage DNA. Rest is from plasmid. Okay, there is one ori of replication, and there is one site for eco R1. So this is how a typical Classmate looks like. All right. Now, so how exactly cloning is brought about in case of Classmates? Now, how cloning is done? The Classmate is opened at unique restriction site. The Classmate is opened at its unique restriction site, and a new DNA fragment is inserted. And then, as usual, ligation is carried out. Now, I have to tell you one more concept, what is known as concatemers. Concatemers, okay, framing of concatemers. This is a new concept what you are going to learn with reference to Classmates. What exactly is done? See, you will cut open the Classmate at its unique restriction site, and you will add the new DNA fragment that is to be inserted. Then ligation is carried out so as to frame the concatemers. What is this? During annealing and ligation of DNA insert, in fact, DNA insert is cut into many pieces. During annealing and ligation of DNA insert, two or more Classmates may join end to end. Two or more Classmates may join end to end, producing what is known as a concatemer, which is the proper precursor for packaging the genome into phage hosts. Okay. Now, if the inserted DNA is of right size, the in vitro packaging cleaves the Cas sites and places the recombinant Classmates in mature phage particles. These phages are then used to infect an E. coli culture. Though the clear plaques are not formed, though we say selection is by clear plaque formation, clear plaques are not formed, then what should we do? Instead, infected cells are plated onto a selective medium and antibiotic resistant colonies are grown. You must be knowing here that there is one ampicillin resistance gene, right? So, that theoretically here one advantage is all colonies are recombinant. All colonies are recombinant because non-recombinant linear Classmates are too small to be packaged into heads. So by rule, if you get a colony, invariably it's going to be recombinant colony. Alright. What are the typical features of a Classmate? They are used to clone DNA inserts up to 45 kb. An advantage. Earlier, it was 25 kb with lambda phage. Earlier, 15 kb with plasmids, but here, as big as 45 kb DNA inserts can be cloned. Then, packaged into lambda particles, which infect the host cells. It is many folds more efficient than plasmid transformation. Transformation is more efficient, okay, in case of Classmates. Then selection of recombinant vector is based on the procedure applicable to the plasmid, which goes into the formation of Classmate. Okay, what type of plasmid you select to design a Classmate will decide the type of selection of recombinant vector. Then these Classmates are amplified and maintained in the same manner as the contributing plasmids. So these are all the typical features of a Classmate. Dear students, this is all about the lambda phage vectors and Classmates. when you are back for offline classes. If further any details need to be given, will be given in the offline class. Thank you for your patient hearing. Thanks. Any questions, you do contact me.